So in a couple of years from now, what is going to be the second leading cause of cancer deaths? Where if you're diagnosed with it, you only have an 8% chance of living for the next 5 years. And it happens to be a major market opportunity, even today. It's pancreatic cancer. Now, Steve Jobs was actually kind of lucky in a way. He had a rare form of it where the survival was measured in a few years. But for most patients, their survival is measured in just months, and some even just weeks. For the 50,000 Americans diagnosed every year, their tumors are like Fort Knox. We just can't get enough of the drug to reach the tumor to have a beneficial effect. So at Vesselon, we have a very lofty aim. We want to double the median survival for these patients. And if we're successful in doing that, not only do we save patients' lives, but for our potential pharmaceutical partners, we can double their revenues without them having to change their drug in any way. So at Vesselon, we don't discover new drugs. We don't even make the drugs. What we do is we make drugs work better. And how we do it is we're taking an old diagnostic contrast agent that's in a mature market, and we're repurposing it. We're changing it to have a higher value therapeutic use for drug delivery. So we intend to file an NDA for a combination drug-drug product that involves an approved microbubble with approved therapeutics. So what are microbubbles? Well, they're injected intravenously, and they're used routinely as a contrast agent for echocardiography imaging. And red blood cells, microbubbles are about half the size of red blood cells, and they stay completely within the vasculature. They never leave, although after some period of minutes, the inert gas is diffused out of and excreted through the lungs, and the lipid shell is processed by the liver. But for drug delivery, we use a different mechanism. We use the mechanical action that's created when ultrasound interacts with the microbubbles, and it creates cavitation. And what happens with the cavitation is that the microbubbles expand and contract very rapidly, and what they do is they expand and contract the vessel walls, and that's a process that temporarily opens up pores to allow drugs to get directly to their target. Next slide, please. Next video. So this is a process that only takes a couple of minutes of ultrasound energy. And during that time, the microbubble cavitation is gently massaging the endothelial layers of the vessel walls to open up these pathways that are temporary, and they're nanoscale size. So it allows much more concentration of drugs to get directly to the target than systemic delivery. It's a process that's referred to as sonoporation. And again, the drugs do not need to be reformulated in any way because the drugs are actually co-administered with the microbubbles. So we're commercializing technology that comes out of Dr. Elisa Conefagu's lab at Columbia University. She is a leading world expert when it comes to sonoporation using microbubbles for drug delivery. Her lab has extensively tested this technology in mice, rats, and monkeys over the last 12 plus years. Research has shown that we can deliver small molecules, certainly, but large molecules, antibodies, for example, we can get 2 to 5 times greater concentration of antibodies directly to the target compared to systemic delivery. So the platform has very broad applications, not only in chemotherapy, but immuno-oncology. So we can get, as I mentioned before, we can get significantly more monoclonal antibody to the target, but it also has been shown to elicit or to activate an immune response. And what that does is it enhances the recruitment and delivery of T cells into tumors. So there's been a tremendous amount of preclinical work that's been done in this space, and clinicians are seeing this breadth and depth of the preclinical data, and they're pushing this technology out into patients. The initial safety studies have focused on very difficult diseases to treat, like brain cancer and stroke. And safety has been shown using, when you add microbubbles, safety has been shown because there's been no increase in dose-limiting toxicities or adverse events. In solid tumors, there was a paper that was just reported last year where clinicians in Norway treated pancreatic cancer patients. And what they showed was when you added microbubbles to chemotherapy, that the patients were able to tolerate significantly more courses of treatment. But more importantly, those patients that were treated doubled their median survival to 18 months. And that gives a strong indication that there's a signal of activity when you add microbubbles. So there are companies that are trying to use microbubbles to enhance drug delivery. These happen to be a couple of companies that are very device-focused. One company, Carthera, is a company that uses a surgical approach. They create a craniotomy in the skull, they implant a transducer, and then they inject microbubbles to augment chemotherapy. Obviously a very invasive procedure, an approach very expensive. And Cytec uses a high-intensity focused ultrasound machine that is a $3 million capital equipment. It's embedded in an MR suite, requires a highly specialized and trained team to operate. Now, for drug delivery, as we mentioned before, it's only a 2-minute procedure to apply ultrasound energy to enhance drug delivery. Columbia's technology is uniquely positioned because it's a portable system, it's very scalable, and it actually is very cost effective. So you're probably wondering, I hope you are, that since people are already using microbubbles today, well, what's so special about our approach? And Columbia's patents, issued patents and pending patents, are very, very broad. They cover the way to optimize microbubbles for drug delivery. But also, we have the only FDA-approved microbubble that's seeking new therapeutic indications. And there are only three other microbubbles that have been approved by FDA. All of them are owned by companies that are exclusively diagnostic. They do not have a therapeutic business nor business model. So second part of our strategy to protect our business is to collaborate with pharmaceutical and biotech companies. We believe that when we team up with them, that they will have a vested interest in protecting their new drug-drug combinations. And then finally, we intend to seek orphan drug designation and status, which obviously would provide 7 years exclusivity, which is a very powerful barrier to competitive entry. Our go-to-market strategy. We intend to conduct a phase 2 clinical trial, piggybacking off of the Norway study. And based on data from that, we expect that we will be able to enter into strategic partnering deals where we would have the companies commercialize the technology for us. And in a drug-drug application, we would use the proceeds from these partnering deals to develop proprietary ultrasound devices that we could use in a device-drug combination to open up the blood-brain barrier to treat neurological conditions. These are all applications we believe that are subject to accelerated review, and together they represent a multi-billion-dollar addressable market. Our team has a collective 80 years experience in ultrasound, microbubbles, and drug delivery. We've successfully built healthcare businesses and raised capital. Our scientific team is led by Dr. Konofagu and Ernie Schutt, who is the inventor of our microbubble and has 59 microbubble patents, clearly an expert. We're also working with Wayne Pambianchi and his group, the SAGE Group, to help us with our partnering discussions. So we're building shareholder value by focusing on repurposing a commodity product, a microbubble, and we're changing it to make it a high-value therapeutic drug delivery tool. And we've raised $1.8 million to date to fund the acquisition of the microbubble, as well as to begin our collaboration with Columbia. We're raising capital now to build our bubble and to obtain an IND, as well as to fund our phase 2 clinical trial. So our customer, we believe, is really the pharmaceutical or biotech company, where if they really want to have a competitive advantage, they can team up with us to deliver significantly more of their drug directly to the target. We can make we can increase the number of indications for their drugs, or even help drugs that have just missed their efficacy endpoints. And all of this without having to change their formulation, again, in any way. So again, at Vesselon, we're not discovering new drugs, we're not making the drugs, we're making approved drugs more effective. Thank you.